Hi, I am Mrs. Sloan, and today it's all about cellular respiration. Now, cellular respiration can be done two ways: aerobically with oxygen, that's how you and I do it, and anaerobically, which we do as backup, but some organisms, that's how they survive. Today's video is going to be an overview of cellular respiration, both aerobic and anaerobically, some of the coenzymes and the process of phosphorylation, and then we're going to focus specifically on anaerobic respiration. This chapter, part 2, is going to be a second video. I'm going to talk about specifically aerobic respiration. All right. So we're going to start with the overview. And you can see here I said deep breaths. Because the most efficient way to do aerobic respiration or to do cellular respiration is aerobically with oxygen. Okay, so on the overview, I'm going to focus on the most efficient way, aerobic respiration, here in our overview. So we eat food, so we can break down the food and release the energy out of the bonds of the food, and we can transfer that energy into ATP. And the best way to do that is with oxygen, and that's why we breathe. So here's the overview or the equation of cellular respiration, and you're going to need to want to know this. Okay, you're going to want to know this equation. So here's glucose, C6H12O6. You can see there's 6 carbons here, and as a product, you still have 6 carbons. It's balanced, right? But we have broken down the glucose into carbon dioxide. In order to do that efficiently, we're going to use oxygen, and we're going to couple this exergonic reaction with the endergonic reaction, the formation of ATP. So to the left here, you have high energy. To the right here, you have low energy CO2. So on your notes, on the basics, you're going to break down carbohydrates to build ATP. Break down carbohydrates to build ATP. Now, I will show you other nutrients and how you can oxidize those as well at the end. So it consumes the gas oxygen, and it produces carbon dioxide. And then I gave you the equation there that you're going to need to know. So let's talk about what gets oxidized and what gets reduced. We've talked about this before, right? Glucose is getting oxidized into CO2, means it's losing energy, losing hydrogens, losing electrons, and the O2 is getting reduced into water. Now, when that happens, you're going to transfer the electrons from glucose, and you're going to transfer it to a energy transfer molecule, a coenzyme called NAD. This is similar to NADP that we used in photosynthesis. I'll talk about that in just a minute, all right? So you're going to transfer them temporarily. So on your notes next where it says energy, it says high energy is glucose, high energy is glucose, and low energy is CO2 and water. Now, let me make myself a little bit bigger here for a minute. I'm going to I know this is the overview, but I know you can handle it, all right? I'm going to contrast this with photosynthesis. In photosynthesis, remember the light reaction? The whole goal there was to excite electrons from the chloroplast, and you transferred that high energy electrons when they went through the electron transport chain, and you created a hydrogen ion gradient, bringing hydrogens to the center of the chloroplast in order to generate ATP. Then we excited those electrons a second time in photosystem 1, and then you reduced your NADP, and then you could use both the ATP and the reduced NADP to form your glucose, to reduce your CO2 into sugar. We're doing the opposite of that. Instead, we're starting with the sugar and breaking it apart, generating CO2, and transferring those electrons from the sugar to the coenzymes NAD and a new one you're going to learn about, FAD. And in that process of doing that, then we will take the NADs and the FADs will drop off the electrons at the electron transport chain of the mighty mitochondria, the bank of the ETC, where we will make ATP. So it is exactly the opposite of what we learned in photosynthesis. So on your notes where it says electron pathway, it says glucose gets oxidized and electrons are passed to coenzymes. Those coenzymes are NAD and FAD, and you have those in your notes. which then drop off the electrons at the electron transport chain, which ultimately reduce oxygen into water. And energy is harvested from the electron transport chain and is used to make ATP. Now, the notes I'm referring to is in the descriptor of the video down at the bottom. I give my students notes. There's 2 columns, so in case I forgot to tell you that before. Column 1 is the scaffolding of the notes that we're filling in now, and column 2, I encourage you to put pictures and visuals. You can pause the video and grab pictures if you want to help you with the learning. So the reactions of cellular respiration allow us to transfer the energy from the glucose into ATP, and it is released slowly. That's what you want to put in your notes. It is released slowly. Therefore, ATP is produced gradually, and it is still only about 39% efficient. Only 39% efficient. And now I want you to look back up in the notes at the first section where I gave you the equation for cellular respiration, and you can see 36 to 38 ATP is crossed out, and next to it you can see 30 to 32 ATP. And let me address that. First of all, remember our laws of thermodynamics. Energy cannot be what? Created or destroyed, right? It can only change forms. And every time you change forms, you lose some as heat. And that's why you only see it being 39% efficient. But when they first kind of were understanding these equations and the formation of ATP, mathematically, it would show that you could generate about 36 to 38 ATP. But remember, it's not even that efficient. You still lose a little bit more of, lose some energy as heat, and that has been course corrected here to maximum of like 30 to 32 ATP. So I wanted you to have that because you will see some diagrams and drawings when you're studying this where it will add up to 36 to 38, but it's really the second law of thermodynamics coming to play, and it's really only about 30 to 32 ATP that you generate, okay? So in case you see that, I just wanted to explain it. All right, so let's start talking about those coenzymes that we use to carry, like the mule, carry the electrons from the glucose into the electron transport chain in order to generate ATP. So we're going to talk about NAD. Very similar to NADP, but here it is in its oxidized state. Here is what it would be. Here is the molecule. And then look here at the reduced NADH. You can see here's a single hydrogen. Here you've added on an extra hydrogen. So it's reduced, it's gained electrons, it's gained hydrogen. This right here is what you're going to take to the top of your electron transport chain so you can na and make ATP. Now, NAD is similar to NADP that we've already learned about before in photosynthesis, right? Here's NAD. Okay, here is NADP. Look right here. Here's that phosphate group, just a little bit different. Now, this points to, right, common ancestry, common biomolecules to be used in energy transfer. That would support the ideas of evolution. Now, a second player in cellular respiration, we use NAD and we use another compound called FAD. Now, if I was going to compare these, NAD is stronger than FAD because NAD, when you take it to the bank of the ETC, you can get a little less than 3 ATP, but when FAD comes into the electron transport chain, he doesn't get to go in at the tippy top. He comes in partway down, and as a result, he doesn't get to take advantage of the full electron transport chain, so he makes a little under 2 ATP each time. So on your coenzymes in your notes, NAD and FAD, each which can accept electrons temporarily, forming reduced NAD and forming reduced FAD. They are not consumed. They are recycled and reused again and again, just like we saw NADP in photosynthesis. All right, now let's look at the big ticket item, the steps. So what I'm showing you right here would be if you were doing aerobic respiration in the presence of oxygen. So if you look at your steps, aerobic respiration with oxygen. Now, the first step in all cells do the first step, which is glycolysis. Regardless if you have oxygen or not, you do glycolysis, and that occurs out in the cytoplasm outside of the mitochondria. The second step, because remember, you're slowly breaking down the glucose. And so the second step, you're continuing to oxidize it, and you form something called acetyl-CoA. And I will show you how that occurs. And acetyl-CoA is within the mitochondria. The third step is the citric acid cycle. Now, the citric acid cycle, if you remember the Calvin cycle, the light independent reaction, where we were building sugar, this is just the opposite of this. Okay, we're continuing to break down the sugar, and instead of taking CO2 and forming it as a solid, we're breaking the CO2 off the sugar and releasing it back out into the atmosphere. Now, all of these steps, glycolysis, the formation of acetyl-CoA, and citric acid cycle, all of these steps, you're generating NAD, okay? And in the case of the citric acid cycle, you're also generating FAD. These are high energy compounds. You're reducing the NADs and the FADs so that you can take them to the bank of the ETC, and it's an electron transport chain that occurs along the inner membrane here of the mitochondria. And you're going to couple that with the chemiosmotic synthesis of ATP. Just put a little something in your head. Remember, because this is all the opposite of photosynthesis. Remember in photosynthesis where we brought the hydrogen ions to the centermost part of the chloroplast and let them go across the membrane to generate ATP? Well, in cellular respiration, we put them in the outermost. So when you look at this mitochondria, this right here where I'm highlighting is called the inner membrane space. That's where you're going to concentrate all your hydrogen ions, and then they will come into the matrix, the center part, in order to generate ATP. That is if you are doing it aerobically. So on your notes where it says steps, aerobic respiration with oxygen, glycolysis, preparatory reaction or transition, that's the formation of acetyl-CoA. The third step is the citric acid cycle, and then the electron transport chain. Now, if you do this anaerobically without oxygen, the first step is still going to be the same. The first step is glycolysis. Who does glycolysis? All cells. But instead of moving into the mighty mitochondria, forming acetyl-CoA and the citric acid cycle, all of this requires oxygen. Without oxygen, you're going to do fermentation. Now, fermentation, there's basically two types of fermentation. You are going to either form lactic acid or you're going to form alcohol and CO2. And today in this video, I'm going to be focusing on the details of that step. So the two most common ways of anaerobic respiration is doing glycolysis, and then you follow it up with alcoholic fermentation or lactic acid fermentation. So on your notes on anaerobic respiration options without oxygen, you see fermentation, and then it says glycolysis and fermentation. If you want to add to that, right, you're either going to form alcohol or lactic acid. Now, I want to talk to you just for a minute. Do you remember back when I told you about that electron transport chain, right? When I showed you the steps? Okay, there are a few organisms where at the bottom of their electron transport chain, instead of using oxygen as their final electron acceptor, in the notes you can see it says, the final electron acceptor at the bottom of the electron transport chain, in some bacteria and microorganisms, they use ions like nitrate or sulfate or even sometimes carbon dioxide to collect the electrons at the bottom of the electron transport chain. And since they aren't using oxygen there, then that would also be considered anaerobic. But those are exceptions to the rules, okay? All right, now, the third thing I want to talk to you about on your overview are just the types of phosphorylation. So right here. So this third one is the review one, all right? These first two are going to be new to what we're talking about here in your mitochondria or in animal cells, at all cells. I don't know why I said that. Sorry, tired. So on the types of phosphorylation, the first one is substrate level phosphorylation. Basically, this is just a handoff. So here you can see it's just passing a phosphate group from one molecule to another. And you see this in two steps of cellular respiration. You see this in glycolysis, and it doesn't generate a lot of ATP, but you see it in glycolysis, and you also will see it in the citric acid or the Krebs cycle. So on your notes, substrate level phosphorylation, passing a phosphate group, and you will see this in glycolysis, and you also see this in the Krebs citric acid cycle. The second way you can phosphorylate is oxidative phosphorylation. And this is what I'm talking about in that final step in the mitochondria, where oxygen is the final electron acceptor, forming water. And this electron transport chain, you generate a tremendous amount of ATP. This is the efficient, best way to make ATP. So on oxidative phosphorylation, it requires oxygen, and the oxygen is the final electron acceptor at the bottom of the ETC, and you see this in mitochondria. The third way to phosphorylate is what we learned about in photosynthesis. The third way is associated with light, right? Remember when the chloroplast and the reaction center chlorophyll A and the electrons got excited and we and PQ? That's what I'm talking about there. That's associated with light and the electron transport chain in chloroplast. So associated with light and the ETC of chloroplast. All right, those are the big ticket items, the big overview of it. And now I specifically want to focus on anaerobic respiration. And the beauty of anaerobic respiration is it's fairly simple. And the first step, glycolysis, is also used in aerobic respiration. So it'll give us a head start there. So the first step, glycolysis, occurs out here in the cytoplasm. So basically you're taking glucose, which is a 6-carbon molecule, and through a series of actually 10 steps, you break down the glucose into 2 3-carbon units. And those 3-carbon units are called pyruvate. So in the process of doing that, you have to get the party started. So it actually costs you money. It costs you 2 ATP to get glycolysis started. So it costs you 2, but in return, you get 4 ATP. So that's why we say you net 2 ATP total, because you spent 2 to get the party started. In return, you get 4. So you net 2 ATP. Now, if you had oxygen, you would go in and do the mitochondria, preparatory, citric acid cycle, and electron transport chain, but we don't. That's why it's grayed out. And in anaerobic respiration, the first step is just glycolysis, and we stay out of the mitochondria entirely. So on your notes, okay, I would like you to go to the basics. It's outside of the mitochondria, anaerobic process, which consists of glycolysis, plus the reduction. We have to deal with this pyruvate. I'll show you that in a minute, to either lactate or alcohol and CO2, depending on the organism. Now, this right here, in the process of glycolysis, you actually make 2 reduced NAD. If we had oxygen, we would take this reduced NAD to the electron transport chain, and the NAD would drop off the electrons at the electron transport chain, and it would get oxidized so it could be reused again in these steps. But since we can't go into the mitochondria because we don't have oxygen, we need to have another way to oxidize this NAD so we can reuse it again in the process of glycolysis. So this is solved by the second step of anaerobic respiration, fermentation, and that's the part where you form the alcohol or the lactic acid. So I'm going to show you the details of that, but for the purposes of the notes, you can see purpose is to oxidize. This is number 2 under basics. The purpose is to oxidize the NADH so it can be reused in glycolysis again, which keeps netting you just 2 ATP. So there are 2 options depending on the organism: alcoholic fermentation by yeast, which produces alcohol as well as CO2, and lactic acid fermentation by bacteria, fungi, and us as backup, which produces lactic acid. So let's look at that a little bit in more detail, okay? This is your Minecraft version of it, the simplest version of it. Now, I'm telling you this is simple because in reality, It's really a 10-step process, which could look terrifying. You're not learning all these 10 steps, so don't stress out about that, okay? Let me show you the simple version, okay? In essence, you take glucose, which is a 6-carbon molecule, and you break it in half throughout these 10 steps, okay? And you end up, if you look right here, here's C6H12O6. When you break it in half, you're going to get 2 3-carbon molecules, and they're right here, okay? Here's C3, and you've got 2 of them, okay? Now, in the process, remember I told you, you spin 2 ATP, but you get 4 ATP back. So we net out, and it's just showing you the summary, 2 ATP. Now, also in the process of glycolysis, you are going to reduce to NAD. NAD, remember, is the mule carrying the electrons for us, which you can think of them like a check, and you're going to take, these checks and cash them out for ATP and aerobic respiration. But since we don't have any oxygen, remember, we're going to need to do the second step of fermentation with these 2 NAD right here that we have formed during glycolysis. But okay, look right here. That might have been a little confusing, but I'll explain it in just a minute. See how we have 12 hydrogens total here? So here, notice we have 2 pyruvic acids. 2 times 4 would be 8 hydrogens, right? So where did those hydrogens and their electrons go? They went to these NAD right here, and that's how we reduced them. All right? So a couple things. I know this is terrifying, but just take a minute and look at it. Here's glucose. You end up breaking this glucose in half right here. And when you break it in half, I want to point out, I know this is really tiny, but do you see G3P right here? Do you remember G3P? That was what we made when we did photosynthesis, right? When we did 3 turns of the Calvin cycle. So put a pin in that. I'm going to come back to that in class. But you keep going. If you notice 2 ATP to get the party started, you reduce 2 NAD right here, and you make 4 more ATP, and this is why we say that you net out 2 ATP. Let's see here, okay? Now, the process of making those 2 ATP is what I introduced earlier in this video. It is substrate level phosphorylation. It does not involve an electron transport chain. It's literally handing the phosphates from one molecule to another. So here on our notes, where it says advantages, this gets you energy really quick because you're just breaking the glucose in half and you're able to generate this ATP. So it's a quick burst of energy for organisms. We do this when things like we're sprinting or when you're lifting weights. It's a quick burst of energy, and we use it as backup for us. But small unicellular organisms, they can survive on 2 ATP per glucose. You and I cannot, though. All right, so let's take a look right here. So the inputs are glucose and 2 oxidized NAD, and we have to invest 2 ATP. Our outputs is we get these 2 pyruvic acids, 2 reduced NAD. And so let's look at the net. We had to put in 2 ATP, and we get out 4, so we're net totaling 2 ATP as a result of this process. Now, if you continue to do glycolysis, you're going to end up converting all of your oxidized NAD into reduced NAD. And then when all of it's reduced, you couldn't do glycolysis anymore, and that organism would die. So we have to figure out a way to oxidize this NAD so we can reuse it again, because this is the step, glycolysis, that generates 2 ATP. This is why you do fermentation. Okay, so here you can see anaerobic respiration. Here's glucose forming pyruvic acid. If you had oxygen, you would move into the mitochondria and do aerobic. We can't do that. So here are 2 options. We're either going to form alcohol or CO2, but either way, when we do this second step, we will oxidize our NAD. So this process is fermentation. So take a look here. Okay, here is glycolysis. You can see your 6 carbons here along the bottom. Remember, I said you had to spin 2 ATP to get this party started. And along the way of doing glycolysis and breaking it down into pyruvate, you'll take 2 NAD and you will reduce them. Okay, you need to do this part in order to generate this 4 ATP, which nets you out 2 ATP, right? So that's the condition of getting 4 ATP, is reducing the NAD. So now you're here at 2 pyruvates. We have no oxygen, so we cannot move into the mitochondria. So there are 2 pathways. Okay, animals like us will take this pyruvate, take the inner pathway, and we will take the pyruvate and reduce it into lactate. And when we reduce it into lactate, we can then oxidize the NAD so it can be reused again for glycolysis. A second option, which yeast and plants do, is they will take the 2 pyruvate and they will convert it ultimately and reduce it into ethanol. Now notice ethanol only has 2 carbons, so you will release a carbon as carbon dioxide. This is why we use yeast to make bread, right? Because the carbon dioxide is going to make the bread rise, okay? So it generates here some CO2. So that hopefully kind of differentiates. The whole reason why you do fermentation is to oxidize that NAD. And I have a little song to teach you about that. All right. So moving on here on fermentation, okay, the input here is glucose, right? The output is either going to be lactate or alcohol, and you get to net away 2 ATP. Now, the advantages, like I said, it is quick energy, okay? Quick energy. Let me make myself a little bit bigger here to help you finish out the notes on fermentation. And it's used in food production like cheese and yogurt and bread rising and alcohol as well. The disadvantages of doing fermentation is it only produces 2 ATP, only produces 2 ATP. You and I, when we do fermentation as backup, when we don't have enough oxygen, if you've been working out so hard that you're like, and you're breathing hard like that, okay, you're going into oxygen debt, and you will do fermentation as backup, but the byproduct is lactic acid, which makes your muscles sore. So oxygen debt, breathing heavy, it is the required amount of oxygen necessary in order to rid our cells of lactate. And lactate is toxic to the cell, it changes the pH, and you need oxygen to oxidize it back into pyruvic acid so you can send it into the mitochondria so that you can use it more efficiently, right? And then the energy efficiency of fermentation is only 2.1%. That means if you think of glucose as a $100 bill, you're only getting $2 for it, okay? There are things called facultative anaerobes. I gave you the notes on that. They can make ATP by either aerobic or if oxygen is present, they will do it aerobically as well, and they can switch back and forth. Okay, now I have a whole song to teach you about this, but I will teach that in a different video. All right, so I hope you're having a great day, and don't forget to watch video 2 so you can learn aerobic respiration.